जब भी हालत है अब मैंने बंद करके रखना होगा अपना कोई तो ज्वाइन ही नहीं कर रहा है नो वन इज बैटिंग मी
कमाल तो नहीं है ना वॉइस जा रहा होगा
हो गया
मेरा नहीं चल रहा
मनजीत कुमार दिखाते रहा
अरे तो मेरे को वीडियो कैसे दिखेगा
ज्वाइन अभी तो नहीं वीडियो आएगा ना अभी वो ज्वाइन मतलब अभी शुरू नहीं हुआ है
स्टार्ट करते हैं Hello everyone, myself Dr. Lokesh Singh Thakur, one of the coordinator of these 3-day online workshops, especially for students on experimental methods in analytical chemistry and advanced chemical engineering. On the behalf of Chemical Engineering Department, Ujjain Engineering College, Ujjain, I welcome you all on the second day of these workshops. Yesterday, 2 lectures were delivered by Dr. Gyan Prakash Sharma and by Dr. M. Chatterjee. We had learned a lot of technical basics and know-how related to the electrochemistry and its applications. Today, we are going to conduct 2 lectures. One from 11:30 to 12:30 will be delivered by Sri Anshul Awasthi. Director and co-founder of Canopy Techno Solutions Private Limited. And the second lecture is from 12:30 to 1:30 from Dr. Gyan Prakash Sharma, Research Advisor, Canopy Techno Solution Private Limited, on solar hydrogen generation, the future of green energy. Now, I heartily welcome today's speaker, Shri Anuj Awasthi. He is the director and founder of Canopy Canopy Techno Solution Private Limited. Before start of his lecture, I want to introduce Mr. Anuj Awasthi. He is a director and co-founder of Canopy Techno Solution Private Limited. He obtained his BTech degree in mechanical engineering from UIT Kanpur in the year of 2014. He worked as a student research associate in the year of 2013 to 14 in electrochemical and reaction engineering lab, department of chemical. He also worked as a process project associate in the year of 2014 to 15 in electrochemical reaction engineering lab, department of chemical engineering, IIT Kanpur. He also worked as a project associate in year of 2015 in nano science and nanotechnology lab, department of chemical engineering, IIT Kanpur. After that, he started their own firms, Canopy Techno Solution Private Limited. In the year of 2015 and onwards, he worked as a director and co-founder there. He designed a low cost solar simulator with C-class solar spectral match. He successfully designed India's first potentiostat. He also prepared a nanotechnology demonstration kit using different nanomaterials. He also worked as a coordinator in Nirman TechMart 2013. He presented research article on flexible fuel ethanol engine in Tech Kriti 2013. He had founding member of Student Activity Center UIT Kanpur. Now I hand over this platform to Mr. Anuj Awasthi to start his talk on water quality analysis. Sir, please kindly proceed, sir.
A very good morning to all of you, and thank you, sir, for the nice introduction. It is my pleasure to be here to present some of our knowledge base which we have collected over the years. We have been working in the field of electrochemistry and especially electrochemical instrumentation, the part which was untouched by Indian industrialists. So, a group of researchers from IIT Kanpur, including me, Mr. Joy Bhattacharjee, he is another co-founder. Our mentors, Dr. Raj Ganesh Pala and Dr. Sri Shiva Kumar, who are the professors at IIT Kanpur, and Dr. Gyan Prakash Sharma, and other team members. We had a vision of making this electrochemical instrumentation and science and engineering a benchmark in India as well as in the abroad. So here we have been practicing electrochemistry in different streams. We have been serving different industries. We have been serving different institutes for their custom requirements as well as. And in that duration, we have developed some knowledge base. We will be happy to share this with you. Today's topic will be, I'm sharing my screen. Today's topic will be, for this particular session, will be related to water quality analysis, and there we will be sharing most of the details which are specific to instrumentation. Just a minute. I'm just trying to share the screen. So can everyone see the presentation? Hello?
Yes, sir, it is
Yes,
visible.
we can see.
Okay, fine. So allow me to start, please. So shall I start?
Yes, sir.
Okay. So the title of the presentation as goes like electrochemical instrumentation, and in this particular electrochemical instrumentation, I'll be discussing about water quality analysis and pollution control. So, how electrochemical instruments have been used in water quality analysis, and they play a very key important role also. Number of scientists and researchers are working in this field. So, in today's session, students and scientists will be benefited by this. Firstly, I will just try to introduce you with Canopy Techno Solutions. We have a product line of electrochemical workstation and a learning kit which we have developed, and this electrochemical accessories. Along with that, we have few service line which includes electrochemical consultancy and training workshop. And the particular areas which we try to cater is energy, corrosion, sensor, plating, lab instrument, synthesis, and energy, with different segments like instrumentation and other consultancy services. And where do we exist is we are the only manufacturer of this particular instrument. We have more than 100 clients in India till now. We have been fully operational since last almost 3 years. And initial customers include ISC, IITs, and ITs, and other reputed institutes and organizations. And whatever the competitors are there, we place our products at least 5 times more uh, cost, less cost. Uh, so this makes us one of the leading particular company in this area segment. Today we'll be discussing about uh, a healthy water, what defines, and I think I try to choose this topic because we also running one particular division in our company in water quality analysis, as well as it seems to be one of the most important topic for now, because the water is very important for the life, and keeping water uh, drinkable and healthy is somewhere our responsibility. So we will see that how healthy water, what defines a healthy water, and some of the electrochemical instrumentation introduction, electrochemical measurement fundamentals we will discuss. And so I will try to touch, I mean, specifically my background is from instrumentation part in the company. So I'll be more touching the instrumentation part. So BTech students, I think, are also there. So they can discuss more and more instrumentation part from my side, so that if they need any kind of insight into this particular field, I can discuss. And then we will be discussing specifically about electrochemical detection of metal ions, where metal ion detection has been done with electrochemical methods very efficiently. Some of the current status and future, I mean, where the electrochemical direction heading towards into the market, and some of the applications. So we will start with a definition of healthy water. So a healthy water means if we take a sample water and we try to get information about what are the parameters which will define the quality of particular water. So I have categorized, I mean, we can categorize into two parts: physical parameters and chemical parameters. Physical parameters, by looking at water sample, we can see that if it is blackish in color, of course there are lots of uh, components are mixed into this, which makes it non-drinkable. If it is clear, transparent, then we will see that at least solid particles are not present there. But still, if water is very clear and transparent, doesn't mean that it is drinkable. The other factor is order. So by small, I mean, smelling it, the order of the water can tell you that what if there is a good quality or bad quality water. Turbidity is, these are the physical parameters. Turbidity is one thing. So I mean, we see that flowing river water, there are small particles. So how much uh, how much transparency is there in the water, and what is hiding that transparency? I mean, some solid small particles are there, sand particles are there. So the measurement of those particular solid particles is called the turbidity. Conductivity is also there. So the part which I told, like there might be some ions which are not visible from our bare eyes, but they are still there. So they are ionic impurities. So by measuring the conductivity of water, we check the ionic impurities mostly present in the water, and the temperature uh, of the water. Another parameters, so these were the physical parameters, and since centuries, people have been judging water on this physical, because what we can judge, this judging the physical parameters is easier than the chemical parameters. So now let's come to the chemical parameters, which is somewhere complex. It needs specific methods for determination. For example, the pH measurement. I think we all of know the pH measurement. We have seen a pH meter in all of our lab. So whenever the pH of water, which is power of hydrogen ion or hydrogen ion concentration exactly. So if the pH of the water goes beyond a particular limit, it becomes non-drinkable. It may harm a particular person or living being if it goes inside. Acidity is one thing. If some so apart from hydrogen ion, there are some other acidic compounds which makes more acidic. And alkalinity is there, so it's the opposite of acidity. And then there are some waste solid parts. So another thing is the hardness. So hardness is again the ions. So these are the chemical parameters which other chemicals mostly which come out from the industry waste. Lots of tanneries are there. So these chemicals directly go into this. And one important parameter is the dissolved oxygen. So dissolved oxygen is one where we actually say that if less oxygen is dissolved, means it is less healthy water. So whenever we try to judge water for specifically for living underwater living species, then dissolved oxygen one of the best parameter. So as we mix more and more carbon dioxide based products into water or waste into water, then its dissolved oxygen becomes less. So that makes it less living. And where we will be focusing on the electrochemical measurements. So this slide actually tells us that why electrochemical measurement of water is or parameters in the water is very important, because most of the parameters you see can be measured electrochemically. This pH measurement or pH meter is a very well-known traditional electrochemical device itself. So it simply has a very good ion selective electrode which measures the pH in that way. In further slides, I will more discuss about it. And the next one is the acidity. We can simply measure by the electrochemical methods. So all in all, wherever ions are involved, wherever ions or electrochemically active chemicals are involved or molecules are involved, we simply can measure with the electrochemical. So electrochemical methods, that's why it makes it very versatile. Dissolved oxygen can be easily measured by electrochemical methods. So there are dissolved oxygen sensors available, and the measurement units, they are traditional electrochemical devices. On the other hand, there are some ions, specific ions and molecules, where we need ion selective electrodes and their related instruments. Chloride and fluoride is being measured by that. Metal ions and heavy metal ions are being measured by that. Nitrates and phosphates are being measured. So before we directly go into like how we can start measuring these things to electrochemical methods, I will be more interested in covering the instrumentation part. So before going to instrumentation, some small introduction of what do we exactly measure in an electrochemical cell or electrochemical system. I believe that most of the fundamentals have been covered yesterday's presentation, so it will be really helpful for you. Any of the electrochemical system includes solution and electrode surface and interface. So we take a particular jar or vessel, we take a particular solution or sample, and then we dip our electrode. So this electrode makes an interface with the solution. So in whole the electrochemical studies, we study these three parameters. And then we start measuring. So once we start measuring and once we are interested to measure it, we can see that there are three basic categorizations which we find. Suppose there is the first one is the colorimetry. Colorimetry can be divided into potential static or galvanostatic. So in colorimetry, we are more interested in measurement of current, you can see. So this ampere meter, you can see, ammeter. So ammeter is required in all the colorimetric methods where we are interested in the charge, how much charge is there. So, and mostly it is used for analyte concentrations. Second one is the potentiometry, where we are more interested in measuring the voltage. So any one of the electrochemical device you take, pH meter, diol, oxygen meter, conductivity meter, one of them will be belonging to present here, colorimetry or potentiometry or voltammetry. So in potentiometry, we mostly try to get the analyte composition. And last one is the voltammetry. In the voltammetry, we try to apply a potential and measure the current. So other two, we were not applying any particular electronic response. And in this voltammetry, we are trying to apply a potential and measure the responses. So here we study the reaction kinetics. So why? In today's session, we'll be more looking into this voltammetric methods. So a broad categorization is there. Electrochemical measurement we do where we do potentiometric measure means we are not controlling anything, we are simply measuring the potential. And when we say potentiostatic, we are more interested in we control the voltage and measure the responses. And when we say galvanostatic means we control the current and measure the potential. So this is just an exercise slide, and I would invite the audience which can involve themselves here. So for example, the pH meter, I hope that all of us might have seen pH meter or might have worked with pH meters. So pH meter is what kind of device? Colorimetric, potentiometric, or potentiostatic? Anyone from the audience?
So potentiometry, because we get to know about the concentration.
Okay, that's nice. So pH meter is a potentiometry because we are simply measuring the potential. We are not applying any potential. We are not applying any flowing any current in the system. We are simply measuring the potential, and actually we are measuring the potential difference between a reference. So in further slide, I will discuss this pH meter once. So it is potentiometric. Once we talk about potentiostat, and the instrument, yesterday's presentations also you might have seen a picture of the instrument, which is called a 3-electrode system. That is potentiostatic. And galvanostatic. I didn't ask because these were more simpler. Now that one more important question. Conductivity meter is one more instrument which we mostly see in our analytical labs. Anyone from the audience can share. This conductivity meter is colorimetric, potentiometric, or potentiostatic?
So colorimetry, because we are getting the value of current. We are not
Okay. So so what we do exactly in conductivity meter, we simply apply voltage waveforms. Okay, and then we try to measure the response as a current. So in this, it is actually potentiostatic. Sometimes it makes confusion because from the instruments, it doesn't make it very clear that whether we are measuring, applying current, measuring potential, or applying potential, measuring current. So this is a clear-cut differentiation. That conductivity meter, we apply potential waveform, and then as of whatever the impedance due to ions is there in the solution, the current flows. So that current we measure. So that's make it potentiostatic. Anyway, you kept a very good effort in this. And there is one more method, impedance analyzer. So mostly we use potentiostatic, and some of the time, sometimes we use galvanostatic also. Yesterday's presentation, Dr. Moitri discussed impedance analysis, which is very important and useful. Now, I think this might be a very good slide for the students also. pH meter. So how a pH meter works, we can discuss here. This pH meter is an example of measuring the pH of water. So what we are interested in measuring, we are interested in measuring hydrogen ion concentrations. So whenever we are interested in measuring the ion, a particular ion in a solution, most of the times you will see a sort of ion selective electrodes are used. So you can see at the bottom there is a pH sensitive glass membrane. So what is the beauty of this pH sensitive glass membrane, that it accepts this is the membrane. So whenever we say membrane means it will be having a pore. So it's pores, and now the pores can be having two functionalities. Whenever you see any porous structure, that can be having two functionalities. One is that it will be having a specific pore size, the size of those small holes, which will allow specific molecules or ions. Another functionality will be there, that it will be having specific functional groups. So it will allow either some particular positive charge or negative charge or particular configuration. So pH sensitive glass membrane is very much selective towards hydrogen ions. So the pore size is very much specific here. So the manufacturers make sure that the pore size is that hydrogen ion, and they are very small because hydrogen ion is one of the smallest. So hydrogen ion can easily go into these pores, and whenever it reaches inside the pores, it simply, the concentration of the ions in the vicinity of the electrode changes, and this particular concentration changes the potential, electrochemical potential at that particular location, which is actually being measured with respect to another AgAgCl. On the other side, you can see the silver chloride. So what happens? This simply gives us a potential difference. So yesterday's presentation, Dr. Gyan Prakash Sharma might have shared the reference electrode slides with you, and I think if you have any query, you can clear now also, or in the second half, the second presentation is from him, you can clear more doubts from him. Reference electrode is very good whenever we want to measure the electrochemical potential of any electrode. So why it is called reference electrode? Because its electrochemical potential is almost fixed. It varies very, its delta change in the potential of reference electrode, electrochemical potential, is very small with respect to temperature, very small with respect to external concentrations and other factors. That makes it very stable for particular applications. So that's how it makes it, we make it a reference electrode. So AgAgCl reference electrode is being used. So whenever next time you go into lab and see a pH sensor, you can see both of these electrodes are actually combined together, and there are two compartments instead of, so you will not be seeing two different electrodes in your pH meter. There is one electrode. Just for understanding point of view, we separated into two. And what happens here, so the potential of reference electrode is very fixed, and the potential of this pH sensor, this glass membrane, the potential at this particular point changes. So what is happening, this silver chloride electrode, which is there exactly, and the outside solution. So what happens, the ionic concentration in this particular vicinity changes, that makes it changing in the potential of this particular electrode. So we simply measure, because this one is fixed, this one is varying with hydrogen ion concentration, we simply measure the potential here. So this was one example of electrochemical measurement of hydrogen ion. Another is the conductivity meter. So as earlier told, that conductivity means we are trying to measure the conductivity. So these are some more fundamental slides, but I'm interested in discussing more instrumentation part into this. So what we do here, we are interested in measuring the ionic concentration. How? Because that makes it that the ions make any solution of water conducting. So more conductivity means more ions. So uh, most of the times, whenever we see there is a RO water, RO system is there in the houses, and the one person comes with a small instrument that is called the TDS meter. So they try to tell you the total dissolved solids in your water. It is actually a kind of conductivity meter only, and it measures the conductivity. So and then by some approximations, they tell you that this is the total dissolved solids. So this conductivity meter, what it does, there are two plates or two electrodes which are dipped, and these electrodes, the distance between electrodes, which is L, and the area of the electrode is very fixed. So whenever we apply any potential across these two leads, so according to the ions present into the solution, the current flows, and we simply measure this, and you can see this I to V converter. So the importance of this instrument, that it can simply measure the current which is flowing there. Now I think there is now some basic knowledge about how these fundamental and the earlier instruments which I talked about were some fundamental electrochemical instruments which we have been seeing. Now once we move towards the more specific analysis, which is voltammetric analysis, which and it is being involved in different ion concentration measurement, and as well as heavy metal ion measurement. So in electrochemistry, what we are interested in, we take mostly, you can see there are electrodes. One of them makes it a reference electrode, and another is a working electrode, which I have represented as electrode. We dip it and we measure something. And what do we measure? We simply measure electronic response of electrochemical activity species. And there are two conditions, with perturbation and without perturbation. So for example, in pH meter, we measure without perturbation, and in conductivity meter, we measure with perturbation. And the next, we'll discuss about the uh, electrode, three electrode setup. I think Gyan or Dr. Moitri, I think in details, she covered this three electrode system. This is a sample three electrode system. I'm more heading towards the three electrode measurement of water impurities or ions. So this electrochemical workstation, it is very versatile because we can analyze any kind of electrochemical reaction into this single instrument, be it from different fields. We can do ion detection, we can do pH measurement with this, we can do dissolved oxygen measurement into this, we can do conductivity measurement into this by making a specific cell setups or conditions. So what happens in a particular electrochemical system? We take instrument and we connect the three electrodes into this. Initially, we can see there is no potential, and then what we do, we simply dip these electrodes into this, and the Fermi level of the solution and the Fermi level. So Fermi level is sort of electronic energy level that it tries to equilibrate. So in order to make the Fermi level of these two components same, some ions partially or fully transfer from electrode to solution or solution to electrode. That gives you a voltage. So this is a sort of potential, open circuit potential we see there. And we further move into electrochemical measurement. The first method which we will talk about is the cyclic voltammetry, because cyclic voltammetry is one of the basic methods where we start measuring the ions or heavy metals in the water. So cyclic voltammetry, I think Dr. Motri has discussed last time itself. So voltage, what happens here in cyclic voltammetry, the voltage scans in a particular direction, and what happens here, after a particular voltage or energy which you are applying, some particular species gets oxidized or reduced. So if it is suppose ferricyanide, and then it goes to ferrocyanide, then it goes to ferricyanide according to the particular potential. There is some, I think, mistake. It should be 4 minus. And I think yesterday she might have discussed that in the negative direction we consider it to be reduction, in positive direction we consider it to be oxidation. So let us discuss some proof of concept experiments. So proof of concept experiments, let us see electrochemical detection of ions, you will see. So for example, we want to measure different ions which are a sort of pollutant in the water, like heavy metal ions. There's iron, more iron, Fe 2 plus or 3 plus mostly present in the water, that makes it more undrinkable. So how do we measure it? So there are other methods which are being used for the detection of ions. So why electrochemical method is very important here? Because it is more sensitive, it is inexpensive, it is fast, and it is portable. In the further slides, I will discuss one very portable system which is being used nowadays. Less sample preparation time it is there. It doesn't use expensive reagent, and we use cyclic voltammetry and differential pulse voltammetry. So differential pulse voltammetry and cyclic voltammetry in yesterday's presentation they were introduced, I think. So firstly, we were using cyclic voltammetry. So, and then for the ion detection, and then we came up with a new method that is called the pulse voltammetry. So in this pulse voltammetry, we apply pulses, differential pulse sort of voltammetry, and then last one is the stripping voltammetry. So stripping voltammetry is a recent method which is going on, and it gives us very good high resolution for the detection of ions, because it can go up to even part per billion. So this stripping voltammetry gives us very good freedom for making handheld devices for the detection of ions. So let us discuss cyclic voltammetry, one which we have already discussed. It is very fundamental. Cyclic voltammetry analysis is very fundamental analysis for studying the concentrations or the interfaces. Differential pulse voltammetry is specifically used for ion concentration measurement, because what happens here, we simply apply pulses instead of linear ramp. We apply pulses. So what happens? Each pulse, whenever there is a high time, so the high amplitude we say high time. So whenever there is a high time, we do not measure the current. We let the non-Faradic current to be settled, that capacitive. So one big problem with in the cyclic voltammetry is that it includes Faradic as well as non-Faradic. I mean, capacitive and non-capacitive both the current. So that makes the particular method to be less sensitive. And the DPV, we apply pulse and we filter out the capacitive region and then we measure it. So one basic thing I just want. So you can see that in this measurement, the effect of charging current can be minimized. So or the capacitive current can be minimized, which makes it highly sensitive. The detection limit of mostly differential pulse voltammetric methods is 10 to the power minus 8 molar. It has reached to almost nanomolar. One example slide is here. So we have, there is one more thing I wanted to share, that in our company, we invite application for the internship, or mostly internship for the students who are interested in doing research projects. So PG and even PhD scholars also sometimes apply. And even during the BTech, students come here and finish their BTech projects here as well. At least 6 months is the time when we resolve. This particular experiment has been done by one of the interns, and the objective was that we wanted to choose some robust electrode. So here we chose gold as a uh, electrode material, and we wanted to detect copper, cadmium, and iron ions in a particular sample of water. So what we did, we took, it is a differential pulse voltammetry of 1 molar, 1 millimolar FeCl3, copper sulfate. Each of them is 1 molar in sodium acetate buffer. So buffer solution is important because we want to maintain the pH as well as some sort of conductivity of the solution. And we found this is a one of the final experiment. Earlier we used multiple electrodes, and then we found that gold is one of the reusable and one of the standard electrode where we could detect these three particular ions you can see. And how do we proceed with the detection? Firstly, we do this kind of fundamental detection, and then we draw a concentration profile. And then this concentration profile, where we this particular calibration, it is called, we also call it calibration curve, where we do differential pulse voltammetry at different concentrations of the different ions, and then we generate this kind of calibration curve. Now, after this calibration curve, this is called a database for some sort of instrument, ion detection instrument. And then once we are ready with this database, now we are ready to detect any unknown sample, which and the range you can see like 100 micromolar to 1000 micromolar we have done. So if any of the sample, any of the water sample comes in between this, we could easily detect cadmium or copper or iron into this, where the detection of copper can be done very well because the sensitivity is very high, you can see. The next one which is there is called the anodic stripping voltammetry. As I told you, stripping voltammetry in the last this slide I told you, stripping voltammetry is one of the most high, the method with the highest precision which is being nowadays used. So what we do in anodic stripping voltammetry, we take we do electrochemical deposition or accumulation of heavy metal at a constant potential. So it is called the constant potential deposition. In yesterday's presentation, you might have heard of chronoamperometry. So chronoamperometry is being used here. So firstly, what we do, we dip our electrode, whatever we have prepared, we deposit the target ions. So it is not only for heavy metals; it is for any kind of metal ion can be used here. And firstly we deposit, and then this is called the stripping or dissolution. So we dissolve those particular ions into solution again. So this is two-step methods. And how is it being used here? This is one of the example slide of differential pulse voltammetry. What we can see here is simultaneous determination of zinc, cadmium, and lead ions in water samples. So it has been taken from one of the publication where people have used, they have functionalized their materials. Now, this is a kind sort of research they have done. What they did, they used carbon nanotubes. So earlier I told that this working electrode, whichever we use, working electrode can be modified with different configurations of nanomaterials, which make it more selective towards the particular ions. So it's easier to detect a single ion, but it's very tough to detect multiple ions from one particular nanomaterial. So if someone can develop a material which that should be electrochemically active also, so that, and the conductive also, because we load these materials on our electrode. So they have taken SP, means screen printed electrode they have taken, and then they have deposited their materials on this, which are based on carbon nanotubes, and then they did this particular differential pulse stripping voltammetry. So what is the meaning of differential pulse stripping voltammetry means? Firstly, they deposited the ion and then they did differential pulse voltammetry. So stripping voltammetry, differential pulse DPSV is the extension of DPV, fine, which makes it more sensitive. So here they did, they are successfully able to detect zinc, cadmium, and lead ions. So further you can see, firstly there is the plane, and then they generated the calibration curve, and then they are able to further detect unknown samples. This is the one process I wanted to discuss. Like this is one process we follow. What is going on nowadays? So earlier, this particular paper, why I chose this paper, because it includes screen printed electrodes. So when we went into market sometimes to solve these kind of problems, water quality analysis problems, so we got some government projects also that we had to detect the water impurities, and we had to judge the water quality, which is a continuous monitoring also. There we found there is a problem that if we use particular electrodes and we simply dip it for a longer time, the contamination goes into electrodes. So the benefit, here comes the benefit with the screen printed electrodes. These are the disposable electrodes. So we might have mostly seen that there is a glucometer sort of. How many of you have seen the glucometer? It might be in most of use home. Glucometer, which is diabetic patients use to measurement the sugar.
Yes, we have seen.
Fine. So most of us see that. So what makes it very versatile instrument? This is one of the electrochemical, or I will say it is a simpler form of potentiostat. So, and it has reached to millions home till now because of its versatility. And what makes it versatile? Because it uses disposable electrodes. What is the beauty about the disposable electrode? Because we simply use it, and at one particular time it gives its results, and then we discard it. So the specificity, and there is no noise available in that because it is not being reused. So screen printed electrode is one terminology where we screen print the ink on polymers or paper. That makes it very low cost also. So in 10 rupees we can get one glucometer strip, and we simply put the blood sample. There it does the chronometry exactly, glucometer. And here in screen printed electrode we put our nanomaterials, and we put